everybody, and welcome to the fourth design review for the Vascular Implant Port team. My name is Caroline. I'm the project leader and project development engineer. Also on our team, we have Annie, who's the program manager, Kostas, who's the manufacturing engineer, and Marissa, who's the quality engineer. Today, we're going to be talking to you guys a little bit about our team objective. We're going to review some of our previous design reviews and what exactly we've accomplished. We're going to give you guys the reasons that we are holding this design review. We're going to review our design, go over our packaging, some of our DV testing, our manufacturing processes and our COGS, our IQ, OQ, and PQ, some commercialization documentation, our changes in scope that we had, and all of our next steps. In order to keep our team on track for this project, we have come up with a team objective. Our team objective is to design and create an implantable vascular port that is desired by the patients and medical personnel to aid in the administration and delivery of chemotherapy drugs. The team's accomplishments from previous design reviews include a design plan, our finalized CAD drawings, some pre-DV testing, our DFM and DFA considerations, an in-depth tolerance analysis, all of our risk analysis documents, including the DFMEA, AFMEA, PFMEA, and our hazard ID checklist. We completed a risk management plan, our bill of materials, and also we got to the point of design freeze. The team is holding this design review number 4, because at the end of this phase, we have completed a master validation plan and manufacturing procedures. We've created a packaging design, labeling, and IFU. We've determined biocompatibility and sterilization tests. We've evaluated our final protocols. We've evaluated design outputs. We've finalized our design transfer and final design work, and we've also updated our DHF. The main goal of this design review is to work towards the commercialization of our implantable port. As a quick review of our port design, our port consists of 4 parts: the port body, silicone septum, threaded ring, and 6 French polyurethane catheter. After considering manufacturability and our design inputs, the team decided that this design was best. After all pre-DV tests were completed, this design was frozen, and the team moved forward, basing everything off of this port design. Our device will be packaged on an HDPE die card of approximate dimensions 5.5 inches by 5.5 inches, and it will be designed to hold the port and catheter separately, as shown in the image below. The die card will then be placed in a Tyvek peel-away pouch. The pouch will be about 6 inches by 6 inches to allow for easy insertion of the die card, while still limiting the die card's ability to move. The pouch will be similar to the one shown below. Shelf box for our device will hold a single die card pouch system. So one device will be in each shelf box. The shelf box will be made out of cardboard and of approximate size 6.5 inches by 3 inches by 6.5 inches. This will hold the pouch securely with some extra space for additional materials such as our IFU or DFU. The shelf box will look similar to the one shown below. The shipper box was designed to hold a maximum of 10 shelf boxes for transit. The shipper will be made of cardboard and of approximate size 16 inches by 12 inches by 12 inches in order to hold these 10 shelf boxes comfortably. The shipper box design will be similar to the one shown below. Included in our packaging design is the label design for the port. The label design is shown below and indicates various things to consumers, such as the type of sterilization used, which is EO in our case, and that it's a single-use device. The team has also put together an information for user booklet, or IFU. This IFU will go over various things such as the product description, indications for use, contradictions, warnings and cautions, possible complications, the implant procedure as well as the injection procedure, any misuses, and the warranty for our product.
One of the most critical components of producing a medical device is ensuring that it's safe and effective. To do this, you need to perform many tests on the device to show its safety and efficacy. When developing our tests for this product, we first developed test methods, then test protocols outlining the sample size and acceptance criteria needed to pass the test, and then we performed the tests and documented the testing results in reports. All of our tests correlate to design inputs, showing that all of our design inputs are met with the device. The first set of testing performed was the design verification testing. This included 5 different tests. All of these tests had a sample size of 300 because they're all attribute tests. This number was determined based on the risk level of the design input that the test was qualifying. The first test was part inspection, where the acceptance criteria was part dimensions and features are within the specified tolerance on the drawings of the parts, and this test passed. Our second test was our puncture and leak test to make sure that our device would not leak and could be punctured many times, and the acceptance criteria for this was no visible liquid drops with 50 psi injections after 400 injections, and this test passed. We also had a flow rate test to make sure that our injections could travel through the port and through the catheter and be delivered to the patient with no obstructions. The acceptance criteria for this test was that the flow rate through the port and catheter must be at least 90% of the flow rate stated by the catheter manufacturer. And this is to make sure that the port and the connection to the catheter isn't restricting the flow through the catheter at all. And this test passed. Our fourth test was a 3-point bend test, where we wanted to ensure that no visible kinks were on the catheter when up to a 15 Newton load was applied. This test passed. And we also performed a peak tensile force test, again where we loaded the device up to 15 newtons and ensured that the catheter did not detach from the port body, which also passed. We also conducted packaging testing to make sure that our packaging remained intact and kept the device safe. For our packaging testing, we performed a peel test with a sample size of 60 because it is a variable test and also based on the risk levels associated with the design input. The acceptance criteria for this test was a force greater than or equal to 1 lived force per inch of seal when packaging was peeled apart on an Instron. This test passed. We also tested a bubble test to confirm that the packaging had no defects or weak spots, and this was to ensure the safety of the packaging. This had a sample size of 300 because it was an attribute test, and the acceptance criteria of this test was no leaks detected, which passed. Finally, we tested the package integrity with a sample size of 60 because it is variable data, and the acceptance criteria is outlined in ASTM D4169 and 7386, and this is a standardized packaging test, which passed. We also had to confirm that the sterility of our device was not compromised and was not harmful to the patient. To do this, we first tested the bioburden of our device after sterilized, and all of the sample sizes for our sterilization testing are based on standards as well as risks associated with our design inputs. All of them also are required to be drawn from 3 separate production lots, so all of the numbers are shown on this slide are the sample size each from all 3 lots. So the bioburden test has a sample size of 10 for a total of 30 from 3 different lots. And the acceptance criteria for this test is less than 1,000 colony-forming units, which passed. We also tested the bacteriostasis and fungistasis with a sample size of 6 from 3 lots, and for this test to pass, it had to have negative growth, and additionally, the 6 organisms chosen for this test must show material turbidity in 5 days to confirm that the test was a good measure, which passed. We also tested the internal and external bacterial indicator. This test is also known as the half cycle and full cycle test. For the half cycle, we tested a sample size of 10 with 1 positive control from 3 lots, and for the full cycle, we tested an N of 12 with 2 positive controls, again from 3 production lots. For this test to pass, there had to be negative growth on the indicator for both the half and full cycle tests, as well as positive growth on the controls, again to confirm that it was a valid test, and this test passed. Finally, we tested the ETO residue left on our device after sterilization, with a sample size of 12 each from 3 production lots. And the acceptance criteria for this test was that the amount of ETO left on the device after sterilization must not exceed an amount that would result in the patient receiving more than 2 milligrams of ETO per day, or 20 milligrams in the first 24 hours, or 60 milligrams in the first 30 days after implantation. And this test passed as well.
Completed biocompatibility testing. For this, we followed ISO 10993-1 and FDA testing requirements. For cytotoxicity, we looked at the device components tested in mammalian cell culture, monitoring proliferation and death in an incubator at 37 degrees Celsius. For sensitization, we used guinea pig maximization testing to follow whether or not the sensitivities would appear once the device is implanted. For irritation and implantation, both were executed with subcutaneous or muscle implants that informed over the biocompatibility assessment of the material components of the device and the device in its final finished form when used in its intended anatomical location. For acute systemic toxicity and subchronic toxicity, the device was implanted in mammalian cell culture and larger animal models to determine the likelihood of either of those toxicities. For hemocompatibility, the device was tested for hemolysis, complement activation, and thrombogenicity. For chronic toxicity, the device was implanted in mammalian cell culture and larger animal models to determine the likelihood for toxicity that would be extremely detrimental to patient health. Further, for carcinogenicity, we wanted to quantify the total amount of extractables and leachables using analytical chemistry methods with the appropriate sensitivity to determine whether or not our product was sound for biocompatibility. All of these tests passed. For age testing, we followed ASTM F1886 and F1980, which is for visual inspection and accelerated age testing. For accelerated age testing, we looked at the triport assemblies that were manufactured in the manufacturing procedure and placed them in an environmental chamber set to 55 degrees Celsius and 20% the relative humidity for a total of 20 days. After the 20 days, the triport was visually inspected and tested as specified in the DV protocol. In the DV protocol, we looked at part inspection, looking at the part dimensions and features to make sure that they're not in incorrect tolerances and no parts are missing. We looked at the silicone puncture, leak, and flow tests for the silicone septum at the top of the port to make sure that no liquid drops are visible, the 50 psi injection and 4,000 injections, and that the silicone septum showed no signs of disintegrating or leaking, and the catheter showed no signs of clogging. Further, we completed a 3-point bend test to determine whether or not the catheter would detach from the port body, or if any visible kinks were arising on the catheter to restrict the flow. Finally, we did a peak tensile force test to determine whether or not the catheter could be detached from the body with less than 15 newtons of tensile force applied. All of these tests passed, and the sample sizes were all 300 because they were attribute tests. For our transit testing, we used shelf and shipper box visual inspection as our starting point, where we loaded 24 shipper boxes onto a pallet, with the minimum number of shelf boxes in each shipper box being 1 and the maximum being 10. The transit testing included products and dunnage for shipping boxes filled with 1, 5, and 10 shelf boxes, and pallets loaded with 6, 12, and 24 shipper boxes. The remainder of the protocol consisted of pouch visual inspection to identify any points of concern or weakness on the pouch contained within the shipper box, a pouch peel test, and a package bubble leak test, much like the packaging protocol, which we tested for the exhibited strength needed to pull apart the pouch in the Instron 5544, where we needed to exhibit a minimum tensile force of 1 pound. For the package bubble leak test, we fully submerged the package and wanted to ensure that there was no evidence of bubble emission through the package. All of these tests passed.
Moving on to our manufacturing process, the simplified manufacturing flowchart shown in this slide describes the basic process flow followed to build the final product. Any item that involves external processes is assigned a part number and undergoes incoming inspection. A new part number is generated for the tested unit, followed by line clearance of the incoming inspection station. The item is then moved to the port assembly station, where any internal manufacturing process is followed by inspection and the new part number generated for the assembly or sub-assembly, depending on the process. When all necessary internal manufacturing processes are completed, the item is moved to the packaging station, where it is packaged with a final part number being generated for it. The same process is followed for all necessary parts, including the part body, threaded ring, silicone septum, catheter, and all packaging materials. All steps necessary to build and inspect the product are captured in our manufacturing procedure document, and the detailed manufacturing flowchart can be found in the DHF. There are two processes in our manufacturing procedure that require the use of assisting devices or fixtures. The first process is the addition of silicone to the suture holes of the port body. For this process, a custom fixture designed to hold the port body in place and an automated silicone dispenser will be utilized. The second process involves installing the threaded ring to the port body, which requires the use of a torque wrench. For both assisting devices, the installation qualification, the operational qualification, and the performance qualification were completed based on the protocols that were created. Any risks associated with these processes has been captured in the PFMEA, and the residual risk found was determined acceptable and no further risk reduction is needed. Transitioning to the cost of goods sold, the following table shows in detail how the total price per unit was determined. From the bill of materials, the material base cost was determined to be $56.5. Assuming a 15% material acquisition cost, the material cost is $65. 30-minute labor time required to build the product at $100 per hour with a 95% yield brings the total cost per unit at $121. Considering the unit lot charge and the gross margin at 40%, the price per unit is $179.4. Lastly, with a $5 sterilization charge per unit, the total price per unit comes to $184.4.
In the final steps towards commercialization, the team compiled a device master record, which indexes reference numbers for the design history file, the router, and the flowchart. In the master validation report, the team looked at each test, where it can be found, what protocol it comes from, and whether it was a pass-fail. This was in accordance with ISO 13485-2016. In order to revalidate any test, the design of the device would have to be considered and tested with whatever new material is chosen to ensure that the materials are still compatible not only with each other, but with regard to functionality and appropriate use. Each relevant test will have to be re-executed depending on the scope of the change and the team's justification. Further, the team completed a transfer form, which outlines the design transfer checklist, highlighting the bill of materials, routing forms, manufacturing procedures, production procedures, risk analysis and risk management, packaging, and other testing that had to be done in order for our final product to come to life. Finally, the design history file is complete with the number of the reference, the title of the document, the revision number, and all summaries for part numbers and equipment qualifications. At the completion of Phase 4, there have been no changes in scope with regard to the design and implementation of Triport in any way.
Although this project is complete for the design control class at WPI, the next steps that would occur on a product development project, such as the Triport project, would be a submission to the FDA for our 510(k) filing, continuation of all of the aging testing, commercialization work with the marketing team, inventory buildup to build up product so it can eventually be sold and distributed, and then also going over the lessons learned on the project to summarize key things that were good with the project, as well as key findings that could be improved upon in future projects.